afternoon, everyone. My name is Pat Cusick, and I'm an account director with the Muscular Dystrophy Association's Healthcare Partnerships team. Thank you for joining us today for the MDA Industry Update webinar titled A Novel Approach Designed to Protect Muscle in Becker Muscular Dystrophy, presented by Edgewise Therapeutics. MDA's Industry Update webinars are programs which provide an opportunity for the community to receive information and updates impacting care and treatment directly from pharmaceutical, biotechnology, and medical equipment companies. These webinars are just part of MDA's education resources. We encourage you to visit the community education resource pages at mda.org. You will find an abundance of resources, informative recorded webinars, and listings for upcoming webinars and educational events. And if you have any questions or need assistance, the MDA Resource Center is available to you by email at resourcecenter@mdausa.org, by calling 1-833-ASK-MDA1, or you can reach out to the Resource Center at mda.org. At the end of this webinar, you will receive a survey link. Please take the time to complete this short survey so that your voice is heard and you can help us create more impactful programming for future events. Now I'd like to introduce our speakers for today. We have Alan Russell, Chief Scientific Officer, and Joanne Donovan, Chief Medical Officer. And without any further ado, I will hand things over to Alan.
Thank you so much, Patrick, and welcome to this webinar where we're going to discuss Sevasemten, EDG-5506, a novel approach to protect muscle in Becker muscular dystrophy. So I'm going to do the lead off, just give you a little background on the company, the studies that we have going, and then Joanne will take it up with the specific results that we have in the clinical studies to date. This is what they call a forward-looking statement. It's really because we are a public company and we are developing Sevasemten, but it is not approved in any district yet. It's in trials, and that's what we're hoping to demonstrate, that it's efficacious. But right now, this is not an approved medicine. Next slide. Okay, so this is a little bit of history. Edgewise Therapeutics, founded in 2017. We're based in Boulder, Colorado, although a number of us, including Joanne, is over on the East Coast in the Boston area. And our goal is to develop novel muscle-targeted therapies. Our number one goal, the original goal, is really to modify muscle injury in the context of Becker and Duchenne muscular dystrophy, and specifically changing contraction-induced muscle injury, which is really one of the earliest hallmarks of muscle damage in Becker. And I'll go through some specifics of that and why dystrophin is so important to muscle protection. From the very, very first day, our goal was to develop a once-a-day simple medicine that was appropriate for everybody and could be used with other interventions. So we're not a genetic therapy. We're a small molecule that changes how muscle contracts, and by doing so, changes how muscle is injured in the context of Becker muscular dystrophy. Just a little bit of landscape background on a topic that is probably very, very familiar to you. Becker muscular dystrophy really can begin at many, many ages in terms of functional decline. Once you start declining, though, that decline is irreversible, so that there's many cases of people functioning quite well for a prolonged period of time, and then there's many cases of people manifesting weakness in their earlier years. But once people start manifesting weakness, and Joanne will give you some background on this, that weakness is progressive. And living with Becker muscular dystrophy progresses, but poses multiple challenges. We don't have all the answers, and it's one of the reasons that we like to listen to the community and you guys to get the real life experience lived by people with Becker muscular dystrophy, because that helps us better design clinical trials and measure things that really matter to you. I already mentioned contraction-induced injury and why it's so important in Becker muscular dystrophy. We believe that protecting muscle fibers from the injury that they pick up when you're active is key to changing the course of the disease. Next slide. Here's all the studies that we have going. Some of them are active, and some of them we've essentially finished and they've moved on to an open label. So the active one, this is the one that we're really excited about and hope that as many people join as possible, is the Grand Canyon study. So this is the largest study that's been run in Becker muscular dystrophy, 18 plus. Joanne will give you more details on that one, but this one is actively enrolling and we're trying to get people onto the study as fast as possible to show whether this compound works or not. The MESA study, that's the next one under, that's the open label extension study. So if you've been on another study with Sevasemten, then you're going to be transferred once that study is completed over to the open label extension where you keep getting the medicine. We keep monitoring how you're doing, and it just gives you access to that medicine pre-approval. Canyon, that is active, so it's ongoing, but it's not recruiting anymore. That's kind of the precursor to Grand Canyon. So this is what they call a phase 2 study. So this is a placebo-controlled 1-year study. We should have results from this study towards the end of the year. We're very, very excited about that one. And it should hopefully inform the Grand Canyon study that's enrolling right now. And then ARCH is the study that Joanne will show you data from. It was the first study that we ran in Becker muscular dystrophy. It's a 2-year study looking at how people do on the compound, safety and function. And we also have some biomarker data that hopefully will be useful to you. Next slide. So I already mentioned that contraction-induced muscle injury is a really important thing in Becker muscular dystrophy. It turns out that some skeletal muscle fibers are more susceptible than the others, and we target those ones. They're called the fast skeletal muscles. It's a detail that's not super important to you, but we try and make these medicines specific so that they're safe, so that you don't get too many side effects, and we really focus on those fibers that are most susceptible to injury when you're active. So we made this compound, Sevasemten, or EDG-5506, that was the original name, designed to protect those susceptible muscles from damage, regardless of the mutation. So it doesn't matter what dystrophin mutation you have, the goal is that Sevasemten would change how those muscle fibers contract, and in doing so, changes how much they get injured. In animal models of disease, Sevasemten protected skeletal muscles from injury and long-term replacement with fibrosis and fat. So if we could achieve that, of course, in people, then that would change the course of the disease, and that's really our goal. And then in initial clinical trials, and we'll show you some data, we show decreases of biomarkers associated with muscle injury and a trend towards functional improvement. So we'll show you some of that data, and I'll tell you a little bit about those biomarkers in a couple of slides. So here is one of our efforts to demonstrate what dystrophin is doing. On the left-hand side here is a healthy muscle with normal dystrophin, and the dystrophin complex plugs into those blue stripes. So dystrophin is inside the muscle, and it links to the outside of the muscle through this thing called the dystroglycan complex. It's a big blob of proteins that bind to the outside of the muscle, and in doing so, that whole complex links the contracting fibers to other contracting fibers, acts as a bridge between those fibers, and you can see that represented in those blue stripes. When muscle fibers contract, they don't always contract to the same extent, and we've shown you an example here where the middle one is contracting more than the outer ones. And in this scenario, you can really see how that dystrophin bridge helps to support the contraction of that middle fiber. It has friends around it that can spread the load. Okay, if you have mutated dystrophin, on the right-hand side, we've just taken it away just to make life easy for the graphic. Now you don't have those links between the fibers. So when that center fiber contracts, instead of being supported by its bridging friends, it has no friends at all. And that causes stress to concentrate along the fiber. And when muscle fibers get stressed, they do tend to break down. This is exactly what we hope to stop with 5506 by changing how these muscle fibers contract. This is everything all at once, if you like. On the left-hand side is a control muscle from a dystrophic mouse. By a dystrophic mouse, I mean it has Duchenne. It's called the MDX mouse. And as that muscle is contracting, you can see it's getting stressed, and those fibers are falling apart. And you can see them falling apart in those kind of blobs that are forming. That's where a muscle fiber has been snipped up because it's become stressed, and it's just crushing in on itself because now it's no longer supported at all. On the right-hand side is the same type of muscle. So again, it's from an MDX mouse. It's contracting, but now we pre-incubated that muscle with a little bit of Sevasemten. And that Sevasemten changes how that muscle contracts and actually dials down the contraction by about 15%. It's not like a huge modulation, but it's enough to take that stress edge off the muscle and stop it propagating into full breakdown. So that while that muscle doesn't have dystrophin, it doesn't have the support from its fiber friends. Changing the contraction just subtly stops the fiber breaking down. So that's what we're hoping to do in Becker muscular dystrophy muscles. Now, when muscles collapse like that on the left-hand side of the video, they tend to leak stuff into circulation. And many of you may be familiar with creatine kinase. It's one of the first biomarkers used on a diagnostic journey to find out what specific muscle problem that you have, right? Because it's often high in the circulation if you have Becker or Duchenne muscular dystrophy. Now, other things also leak out of injured muscle. And I've put 4 examples there, well, 3. Myoglobin also comes from muscle. Fast skeletal muscle troponin, that's a lot of words. It's a muscle protein that's specific for those susceptible fast muscle fibers, and then a lot of other stuff as well. So we have used these biomarkers to just give us an early idea about whether the compound is protective or not. If it changes how the muscle fiber contracts and the fiber isn't falling apart, you should see a decrease in the leak of these proteins. At this point, what I'm going to do is hand over to Joanne. She'll give you an idea of the clinical studies and the results that we have to date.
Great. Thank you, Alan. And thank you, everybody, for joining. It's a pleasure to speak with you today. What I'm going to talk about is a bit of what we have seen already in the completed studies, the phase 1 study in healthy adults and adults with Becker, and also the ARCH study, the 2-year open label study, and then talk a little bit about the studies to come, basically, that are ongoing at this point. So on the next slide, what do we find out from the phase 1 study? Now, we start a phase 1 study for basically the first time in people. So we're looking very closely at people to understand safety, to understand how the drug is absorbed. So there we saw the drug was well tolerated, check. And how can we give it? We give the drug as an oral tablet once a day, and it's well absorbed whether you take it with or without food. So that's kind of sets the landscape for the future studies as well. And we asked a very important question, whether the drug, the Sevasemten, can reach the muscle. Because if it doesn't get to the muscle, then it's not going to potentially work very well. And what we found is that the drug is highly concentrated in muscle. It's 100 times more concentrated in muscle than it is in the bloodstream. And that tells us it's getting to where it's going to be. It's also something that tends to be very good in terms of safety, that the drug is concentrated in the place where it's working. It's not concentrated in other organs. It's almost exclusively in muscle. So on the next slide, we then went to longer-term studies. And in any kind of a longer-term study in patients with Becker, with any disease, what we want to figure out is what can we use to measure disease changes over time? I mean, you know, things change over time. How do we pick something that effectively measures things in a way that we could do a clinical trial to show whether a drug works or it doesn't work? And what we are using here is something called the North Star Ambulatory Assessment. It has 17 different activities that measure function in ambulatory men. And we score each one, whether it can be completed the usual way or compensated, so that, for example, you're using something to help climb stairs, or not at all. And we go up to a score of 32, and it's been well established. This shows the particular items that are in there. And they were chosen originally for Duchenne muscular dystrophy, but they're very much things that are translatable into everyday life. It sounds like things that, are, more extreme, hopping on leg, being able to get up from the floor. I mean, hopping is something that maybe one doesn't do in daily life, but at the same time, for adolescents, that's important for them. Getting up from the floor after if somebody falls, to be able to get up without assistance is truly important in everyday life. Climbing a step, standing on one leg, to put clothes on. These measure a range of activities that we can see how things change over time. So it's scored, as I mentioned, from 0 to 34. And on the next slide, it very much correlates with what's going on in the muscle. As Alan said, that the muscle is injured when contraction occurs, and over time that builds up, and the muscle is response to that continuing damage is to actually replace the muscle with fat and fibrosis. And that's the big problem, because if you've lost muscle, then you lose function, and the muscle doesn't come back after that. It turns out that it isn't so much about age, but if you've started to lose function, that predicts that you're going to continue to lose function. So the North Star top is 34. If you're there, people may very well be there for a number of years. But once you start to decline, you continue to decline. And what that correlates with is fat infiltrating the muscle. We actually look at that in our studies with MRI. And we know that individuals in this range below 32 continue to drop over time, about 1.2 points, which doesn't sound like a lot, but at the same time, over 5 years, over 10 years, it's a real impact in terms of the way of function in daily life. So on the next slide, the other thing we measure in trials, because we want to see things that change fast, that can indicate something improves in the long term, as well as the function, which may take a while to observe in a clinical trial. So we also look at these muscle protein biomarkers that Alan talked about, because there's something that's relatively easy to measure. You can draw blood and be able to see that these are leaking from muscle, and we can measure them at multiple time points during a clinical trial. So it's something that we look at very clearly to identify whether a drug is working in the way that we anticipate it's working, and to help do things like choose a dose, as you'll see. So on the next slide, what we saw in the ARCH study, now this was a dozen men, open label study, meaning that everybody got drug and they knew they were getting drug during this 2-year period. These were men some of them had been in the phase 1 study, and some additional, so for a total of 12. And what we saw was these biomarkers, the creatine kinase, fell rapidly within the first month, and they stayed down. Now, the y-axis, the vertical, is the different biomarkers, creatine kinase, fast skeletal muscle troponin, and myoglobin. And this shows over the months of treatment from up to 2 years. And the different shades of blue indicate different doses that we looked at. So we want to make sure that we get the right dose for a pivotal study for registration. And what we saw was that we saw pretty much maximal decrease in these biomarkers, even at the lowest dose. So even though we went up, we didn't see more benefit from more drug around. And you never want to use more drug than you need to. So we went back to the 10 milligram dose, and we saw that the level was essentially the same for all 3 biomarkers. The TN and I2 may be a little bit more, a little more variation. So that gave us a lot of information about the dose. So then on the next slide, what happens to function? And we looked at the North Star over, every few months when they came in, and need to give a gold star to these men in their 30s on average. And they came in, it turned out they logged 190,000 miles in air travel, getting to the site back and forth again, where Dr. Han Fan was. And there's a lot of work that went into this. So over the 2 years, what we saw, so this is the change in North Star. Up is good, down is not good, over the 24 months. So what we saw was that on average, these men basically were stable, which is good. Stability is good if it means that you're not losing functions. And one would have predicted from the natural history studies that are following patients with Becker over years, is that they would have over 2 years lost about 2.5 points in their North Star. So that's very encouraging. It isn't definitive information because this is an open label study, so everybody knows they're getting drug, and we don't have a placebo control, which nobody likes placebo control, but from the regulator's point of view, they really want to see that difference emerging. So this is very encouraging. It gave us the information to be able to move on to the next important study. So the next slide, it basically, to summarize, we saw good safety. I didn't show the safety in detail, but it was very well tolerated at all of the doses. We saw decreases in biomarkers, as I showed you, that gave us a dose. And we saw stabilization of function, which is important to folks. If you can hold on to what you have and not lose any further function, that's a win. And we've identified the dose for a pivotal trial to go to regulators. And we have then now started, as Alan mentioned, the Grand Canyon study. And that study is a pivotal study, meaning this is a study we can take for approval, 18 months long, to be able to see changes and look at the effect of Sevasemten, not just on biomarkers, but importantly on functional measures over the 18-month period. So we are enrolling men between ages of 18 and 50 who have a genetic diagnosis of Becker. So you have a mutation in the dystrophin gene. And they are ambulatory and can complete the 100-meter timed test in 200 seconds or less, and that can be with assistive devices, meaning a walker or cane. Our goal is to enroll 120 patients that at about 50 sites across the US, Europe, and actually beyond. In the design is shown on the next slide, in that people initially come in for screening. They're randomized 2-to-1, so that 2 get Sevasemten versus 1 with placebo. And over the 18 months, there are 7 study visits over the entire time. And because of the safety we've seen, we're able to spread those out. So somewhere between 3 and 6 months after the first month. We look at functional assessments. We look at blood tests for safety and MRI, because it's a valuable way of looking at the structure of the muscle and how that's changing. We're not biopsying the muscle, but we're looking non-invasively with MRI. And that's just at the beginning and the end of the 18-month period. So we're doing this on the next slide at many sites through the US. We don't want to just have, we want people to minimize travel. And we are supporting travel, of course, in terms of a concierge level group that is supporting that and prepaying expenses as much as possible prior to the travel. So to summarize, on the next slide, we're developing Sevasemten for Becker. We're also, I didn't focus on it today, but we're also in a dose ranging study to develop it for Duchenne muscular dystrophy. So this is an oral drug to preserve, to potentially improve function in individuals living with any mutation in the dystrophin gene with Becker or Duchenne, and to prevent damage to those muscle fibers that are most susceptible, the fast muscle fibers that Alan spoke about. We see this as being able to be used alone or potentially in combination with other therapeutic approaches, because if you are protecting the injury that occurs with contraction, then that we know that continues despite other therapies, for example, with Duchenne. So we want to be able to use it alone or in combination. So we are, as we started out by saying, we want to listen to the community to understand trial burden and how we can minimize that in the trial. We're also including patient-reported outcomes in the study and doing things like interviews, exit interviews, to understand what the experience was and what the experience of patients in the trial is on the drug, and understand that better, things that we may not have been able to measure with the standard assessments that we are looking at. I mentioned the support services to ease participation burden on families, because it's not just the individual, it's the whole family that is really has a burden. More sites is good. And lastly, to summarize, Becker's a serious dystrophinopathy. There's no doubt about that. And while declining function can begin at different ages, once it does begin, individuals with Becker are on a path that is irreversible to losing muscle and function. And we want to stop that. We want to halt that in its tracks. We know that Becker poses multiple challenges to those living with it, and want to listen to you. And so please speak up, ask questions. We are targeting what we believe is a key aspect of the disease process, the contraction-induced injuries. And we believe this approach can slow the functional decline that occurs on an ongoing basis in those with Becker. So thank you so much. We're happy to answer questions. Here's some contact information, some social media places to follow us. And with that, Patrick?
Yeah, no, I was going to say thank you very much, Joanne and Alan, for the informative presentation. If anyone has any questions right now, you can go ahead and type those into the Q&A window, and we'll go ahead and see if we can answer those. Let's see. We have one here. Any upcoming clinical trials for 10 to 13 beyond Duchenne/gene therapy patients?
Yes, you're correct. We are enrolling those studies. We in the Canyon study, the first part of the Canyon/Grand Canyon, we enrolled adolescents, 12 to 17. There is less known about the natural history in adolescents and children with Becker, and that's something we're trying to gain more information on, and ultimately, and the younger boys as well, the 10-year-olds. We hear you and take that home. We've got to figure out how to approach that as well. So we're getting information from the Duchenne studies in terms of age and understanding absorption of drug, things like that. So we can ultimately get to the whole spectrum of patients with dystrophinopathies. The cardiac restrictions, we ask that patients for inclusion have greater than a 40% ejection fraction.
And then I'm not sure if you saw the next one. Does it stop progression or just slow it down?
We are hoping to stop progression. That remains to be seen in the clinical trial. I think that if you take a disease that progresses over 20, 30, 40 years, and you slow it down and it progresses over 100 years, that's a real improvement. So we are looking for anything that will ultimately improve the lives of people living with Becker. There's information on this slide. You can go to our website or studies at edgewisetx.com. We are also there on clinicaltrials.gov. That points you in the right direction to get in touch with somebody who can help as well. And thank you for listening. Thank you to those who have participated. It's a lot to participate in clinical trials. And we very much appreciate your efforts. It takes a village to do this.
There was a question, Joanne, about a new site in Chicago.
Yes, there is a new site that will be opening or is open in Chicago. Yes, at Northwestern. When we need to enroll this study and get the information. So it does take time, unfortunately, to be able to see the difference and to be able to present a compelling picture to the FDA.
Mm-hmm. There's a question about will the therapy be effective for non-ambulatory patients?
This is it. The disease progresses. The disease is in upper extremity, in the arm muscles as well. We see this when we meet patients. I mean, being able to overhead reach is a clear issue. And so the same process that affects particularly the leg muscles by the injury of contraction also occurs, but more slowly in arm muscles. So we think that this is a general process, and this should be able to protect. We are, in fact, measuring upper arm function as well in the study.
Gosh, so many questions. When will you have and provide results from Grand Canyon?
Canyon should be at the end of this year, and Grand Canyon, we have to enroll it first, and then it will take a year and a half or so after that.
I like this one. Is it possible to request the drug and not the placebo?
Unfortunately, no, because that's the concept of a randomized trial, so it doesn't bias the results if you know that you're on drug or placebo.
Now, if you're on the Canyon study, for instance, and you reach the end of that 1-year treatment period, while you could have had a placebo there, everyone, when they go to Mesa, will go onto the active drug.
Yes.
Okay, so you're not on a placebo forever.
Mm-hmm.
If Grand Canyon goes well, is it possible you might apply for early release of the medicine?
We will do everything we can to get it as quickly as possible. That I can assure. And people have asked about older patients as well. And the reason that we looked at this age range is that there is much more natural history to look at in that age range. And when you're designing a clinical trial, you want to make sure that you understand what's going to happen in patients that are not on drug, the placebo group. And since there's so little information that was available over 50, and there's other things that can change your North Star and function above 50, that's why we did not include those patients. But we do anticipate that it would be helpful in if it's helpful in the younger folks, it should also be helpful in those over 50 and the non-ambulatory folks.
There was a specific question from Randy about that, who's 62.
Mm-hmm.
And then from Kelly, my son will be turning 18 in July. He's diagnosed with Becker. Do you anticipate Grand Canyon enrolling to be ongoing so he might be able to participate?
Yes. And in fact, you can get in touch with a site before then and plan to be part of that. So.
I missed one. When this medicine is available on the market, which of course is dependent on trial results, will this be a therapy be effective for non-ambulatory patients?
We think that the mechanism would apply across the spectrum, and that of course then depends on what the FDA would say in terms of what group of patients would be eligible for the therapy.
And then there's one final question from Anonymous. What's our best estimate of when this treatment will be available on the market?
So when we finish enrolling this study, the process is that then you take the 18 months, then we need to analyze the data and submit it to the FDA. The FDA typically takes between eight months and a year to review the data. So it is a while still.
Yeah, it's a great question, but the only thing that we can control is how fast we get people onto the study. So the minute that we have everyone on Grand Canyon, then the clock starts on reading results and getting that data to the FDA, right? So the current estimation is probably early next year we'll finish recruitment, but like if we can get it quicker, then we can get that data to the FDA faster.
Perfect. All right. Any other questions?
That's great. Thank you for all the questions.
Thank you so much for the great questions.
Yeah, great questions. Thank you, everyone. And thank you all for joining us today for today's industry update webinar presented by Edgewise Therapeutics. We hope you'll join us again in the future for future webinars. And I hope everyone has a great day. Thank you for joining us.
Bye. Thank you.
Thank you. Thank you, Kathy.